Did you say transcription or prescription? I don't know. All right. This is Norris Teaches Science, the Biology Edition video starring me, Mr. Norris. And today I present DNA transcription. Video goals for today is I can explain the process of DNA transcription and why it is important. Hopefully you have watched the other videos on DNA structure and RNA, and you're ready to talk about protein synthesis. And that's where DNA transcription comes in. So, what is DNA transcription? DNA transcription is the process by which DNA is used as a template to make RNA. In shorthand, it's DNA to RNA. That's what it is. And to be technical, it's to mRNA because it's going to be the messenger RNA that it's going to make. So where does transcription happen? It happens in the nucleus. Why? Because DNA cannot leave the nucleus. DNA is stuck in the nucleus. If it leaves the nucleus, it can be damaged. And we do not want damaged DNA. We're going to do a whole video on damaged DNA, and damaged DNA could actually be bad. Or it could be good, but right now we're going to say it's bad. So it's going to happen in the nucleus. Now, why does DNA transcription have to happen? One, we got to get the information from the DNA out of the nucleus to the ribosomes to do protein synthesis. So to do this, we got to make RNAs, which involves making the 3 types of RNAs we talked about in the RNA video. The mRNA for the message, the tRNA to bring the amino acids, and the rRNA to be the ribosome. So we need to make RNA to have protein synthesis so we can get the information out of the nucleus so it can go to the ribosome. So these are the general steps. So the first step is the section of DNA that needs to be transcribed is untwisted and unzipped. We will not transcribe the whole DNA strand like we did in DNA replication. We will only transcribe the section we need, the section that contains the information for the protein we're trying to make. If we're trying to make the protein to be the enzyme to take out lactose, or the protein that's going to determine your hair color, or all the other proteins out there. We're only doing the section that we need. After that happens, enzymes are going to bring in complementary RNA nucleotides, the ones that match up with the DNA strand. And this is how it works. If the DNA strand would say A, RNA is going to say U. If DNA says T, RNA is going to say A. If DNA says C, RNA is going to say G. And if DNA says G, RNA is going to say C. Remember, DNA has T's, RNAs have U's. So we're going to match them up. Once they're matched up, they're bonded together, the DNA is released from the RNA, is released from the DNA. The DNA goes back to its original shape, and the RNA is free to leave the nucleus to go where it needs to go for the process of protein synthesis. So let's practice this real quick. So I have a strand of DNA that we need to transcribe. So we're going to transcribe the strand into RNA, or the complementary RNA, mRNA strand. So if DNA says A, mRNA will say U. DNA says T, RNA is going to say A. If DNA says C, RNA is going to say G. Another C means another G. If DNA says G, RNA is going to say C. And again, what would A go to? I'm hoping you said U. And that's how you would do transcription. This is one part of protein synthesis. The other part we're going to cover in another video, and it's called translation. But we're going to stop here, and hopefully that you can practice this a little bit before we get into translation. But like I always say, this is Mr. Norris. Hope you learned something, and I'm out of here.